Biology, Chapter 1.1. What is science? Covered in the text, page 4 through 9. What is science? Science is an organized way of gathering and analyzing evidence about the natural world. We call these human constructs ways of knowing. Science also refers to the body of knowledge gathered by scientific studies over the years. In addition to science, humans have developed other ways of knowing. In fact, these are much older ways of knowing, including such human endeavors as philosophy and religion. Now, science is a unique way of knowing. It's different from the other ways of knowing that humans use to try to describe the natural world. First of all, science deals only with the natural world. It does not deal with supernatural agents or deities or factors and mechanisms outside of the natural world. Secondly, science involves the collection and organization of information in an orderly way, looking for patterns and connections. Other ways of knowing, such as philosophy and religion, don't always involve collection and organization in an orderly way. Thirdly, science proposes explanations based on evidence, not belief. Religion is a way of knowing that values belief over evidence. So this is another difference between science as a way of knowing and other ways of knowing. The goals of science include providing natural explanations for events in the natural world and understanding the patterns in nature and being able to make useful predictions about natural events. The image on the slide is the projected paths of solar eclipses from the years 2061 through 2080. So without a complete understanding of the relative movement of the Earth, the Sun, and the Moon, it would be impossible to make these kinds of predictions. Science also deals with change and uncertainty. Scientific ideas change as new evidence is discovered. As a result, science rarely proves anything absolutely. Even though you may hear or see in the media descriptions of scientific proof of one idea or another, generally we talk about science providing support or finding evidence for a particular idea. But it turns out in science it's much easier to disprove something than it is to prove something. So the idea is, with this change in uncertainty, that scientific ideas reflect the best understanding of the natural world that current knowledge and methods can provide. And if new information, better information is discovered in the future, then the scientific idea has to be flexible, has to be changed, and or possibly discarded. What is the scientific method? This is a structured way of investigating and evaluating questions. Depending on which reference you look at, you might get different steps, but this is a fairly good representation and goes along with the textbook. So the steps are observing and asking questions, inferring and forming hypotheses, designing experiment, collecting and analyzing data, and finally drawing conclusions. Observing and asking questions involving noticing something that leads to a question. We tend to think of observation as a visual process, but in the scientific sense, observations can be made with any of the five senses. The next step is to infer and form a hypothesis. Just for a little bit of vocabulary, an inference, it's a logical interpretation based on current information. So here's an example of an inference. I'm outside. I notice that it's getting cloudy. I notice that the wind is picking up. I notice that the temperature's dropping. I can infer from these facts or observations that it might be getting ready to rain. So that would be an inference I would draw from evidence in the environment. When you tighten up your inference into a testable form, then you've got a hypothesis. So it's a testable explanation for a set of observations. The experiment. This is also a unique sort of approach to science. Many other ways of knowing don't do experiments. They're based on observations and other processes. But the experiment is designed to test the hypothesis and to determine whether or not it should be accepted or rejected. If possible, when you do an experiment, it should be set up as a controlled experiment. And this means that it's designed so that only one variable is changed. If the design is such that more than one variable changes, it's impossible to tell which variable caused the results that you observe. So the example here on the slide shows an animal behavior experiment looking at the movement of insects in response to wet or dry environments. Now notice that all the possible variables, they're all the same. The number of bugs is the same, the temperature is the same, the filter paper is the same, the light is the same. These are all controlled. The one variable that is different is the moisture, whether or not the paper is wet or dry. And so this is the nature of a controlled experiment. All but one of the variables is held constant. The independent or manipulated variable is that single variable that the experimenter deliberately changes. In the previous example, that would be wet versus dry. The amount of moisture would be the independent variable. When you graph this variable, it always appears on the x-axis. And in this example, plant growth investigation, the independent variable is actually the color of the light. So red versus blue versus yellow, that is the independent or manipulated variable that the experimenter deliberately changes. The dependent or responding variable is the variable that is observed and measured as it responds to the independent variable. In this particular example of plant growth investigation, it is the growth of the plants in response to the color of the light. When you graph this variable, it should always be placed on the y-axis. Here's a summary image showing these variables together. So the independent variable, the dependent variable, and the various controlled variables. So the liquid used to water the plant is the independent variable. The height of the plant is the dependent variable. And everything else that could be different but is held constant is controlled. The type of plant, the size of the pot, the amount of liquid, the soil type, the temperature, the amount of light, etc., etc. You should also provide, if possible, a control group when designing an experiment. And a control group is exposed to all the same conditions as the experimental group except for the independent variable. So in the example of plant growth that we saw earlier, just using plain water would be a form of control. Instead of using soda or orange juice, plain water would give you a control group. That way you have something to compare your other results to. After the experiment is complete, it's time to collect and analyze the data. Qualitative data have no numbers, so things like larger, slower, better, smaller, these are all qualitative descriptions. If the data involves a measurement with a specific number or value, then that's called quantitative data, such as 55 centimeters, 25 degrees Celsius, 3.5 feet. These are all quantitative measurements. To collect data, oftentimes tools are used, such as microscopes or rulers. And when you're collecting and analyzing data, need to be aware of possible sources of error. The last step of the cycle of scientific investigation is drawing conclusions, where you interpret the data, summarize the results, accept or reject the hypothesis, and consider ideas for future research. So in the cartoon example here, we've come to the conclusion that pollution negatively affects fish growth, but an idea for a future study would be, I wonder which chemicals specifically are responsible, which sounds sort of like a question, which is not unusual because actually the process of scientific investigation is often cyclical. There's a nice diagram in the text. Here's my version of it. The cycle, starting from the initial observation, the gathering, the hypothesis, the experiment, the results, the conclusions that can feed back into another cycle of investigation. If you have any questions, please feel free to contact me or jot down some questions and have them ready for our next class meeting.